species, but by the same way, and use the distributional data to find the so-called phyloregions. I will not go into details to that, but I would like just to. No. Je wouldn't sound exactly on the So I just would like to show you this bias on my favorite example, which are the root rats. These are subterranean rodents in Eastern Africa. And before we started to work on them, there were 12 species or 13 species described. One of them, very big one in Ethiopian highlands, only in Bale Mountains, in one mountain range. And then there were 12 species of smaller root rats, one of them in Ethiopia, and 11 species described in Albertine Rift Mountains and Kenyan highlands in former British colonies, because they were all described by British taxonomists, and there were only one species in Ethiopia. But when we performed the molecular genomic study, so we discovered that there are 6 lineages, and this big one is only internal lineage of the small ones, which is quite interesting. And 5 of these 6 lineages, they live only in Ethiopian highlands, and 1 is widespread in Kenyan highlands and Albertine Rift Mountains. So it really shows how biased can be our knowledge on biodiversity, because it completely changes the pattern. So this was the original state, so 2 Ethiopian species and 11 non-Ethiopians. When we look at this genomic pattern, so we have 5 Ethiopians and 1 non-Ethiopian. So it completely reversed the pattern of species diversity. And it can be very similar in many other groups of rodents and other mammals and animals in general. So why there were not these 5? It's very simple, because Ethiopia has never been colonized, and Ethiopian taxonomists have not been very active in describing biodiversity in their unique country. So we went to the field, and we collected small mammals in last 20 years. And many other samples for various purposes. Sometimes it's a big pleasure, as you know, to be in the field, to collect rodents and to see the animals. Sometimes it's less, because there are some problems as well. And in last 20 years, we performed about 30 expeditions to Sub-Saharan Africa mostly. And we have now in our freezers about 15,000 of small mammals, and we collect various types of samples, and you are very familiar with that for various purposes, for genomics and microbiome and parasites and so on. And but if we would like to get some more continental view on diversity, we have to collaborate, and we like to collaborate with other people, including many of you. So we organized with Jonas Meyer to this meeting on African Small Mammal Symposium in 2019, and we continue the collaboration with many people. And we try to get together all the data, genetic data on small mammals. So these are the localities from where we have at least some genetic data from at least one specimen in Africa. So, Madagascar is not really Africa. Sorry, Gauthier. And of course, there are still some gaps. So, for example, we went last year. So this is our last year expedition in Eastern Central African Republic, and we went also to Angola. So we filled some of the gaps, but still, of course, there are some places where it would be good to go and to get some data. So for most of this material, we use cytochrome B as the primary barcoding marker, and because everyone knows that cytochrome B is not can also provide biased information. So we use other markers for particle groups. So we have a lot of DDRAT sequencing data, and now we focus on these ultra-conserved elements, which are about 4,000 loci, and we have this information for almost 1,000 of small mammals. And we complement this by morphometry and any other ecological data, so to perform the revisions for many groups of African small mammals. And so, of course, the first question is how reliable is this large cytochrome B data set, now about 25,000 of cytochrome B sequences on African rodents. And I will not go into detail, but when we compared all these data with cytochrome B data, so it works quite well. So we can sleep well. Cytochrome provide really reliable information in most situations. Of course, there are some problematic groups like gerbils, we discussed yesterday. So for gerbils, the standard cytochrome B barcoding doesn't work. And there are also some places like Ethiopian highlands where there could be a problem with cytochrome B. So just two examples how it can be used. So you can discover new taxa by this barcoding. This happened to us in forests of southwestern Ethiopia. There are very endangered ecosystems, very unique. And by chance, we discovered one strange cytochrome B, and we found later on that it's a new genus of mammals. So we performed many genomic analysis and morphometric analysis, and we described a new genus of Praumini rodents that diverged from other Praumini 6 million years ago. And it's not only from this single specimen, and it's really the place which is highly endangered. And it's in Ethiopia, which is the country with the most intensive growth of human population. So it's really endangered. And now we have a project with Rainforest Trust to somehow create a protected area in this forest, because it was not protected at all. And there can be many other very interesting organisms living in this forest, which is unknown. So then, of course, to make taxonomic, to clean the taxonomy of rodents, it's good to have the information, some genetic information from holotypes. So we also use this museomic approaches. So we sequence the low coverage genomes of all the museum specimens, including many holotypes. This is one example of Dendromus mystacalis, which is a well-known name used for many Dendromus across all Africa. And we analyzed in detail one clade of Dendromus. These are genomic data. So we discovered that there are 7 gene pools, and we needed some names for these gene pools. So we had morphological data. So they are somehow distinct also by external morphology and skulls. And this is the holotype of Dendromus mystacalis without skulls, and collected in 1863. Deposited in Stuttgart Museum. So we were able to get a tissue from this holotype, and we performed the sequencing. We assembled the complete mitogenome, and we know that Dendromus mystacalis is this one. So it's this species. We know that it's distributed in lower elevation of Ethiopian highlands and also in one mountains in northern Uganda. And so we can use this name for well-delimited species. And so this is what we are doing now. We are trying to do in other groups of rodents. So to sum up, we published about 60 papers in last 15 years about taxonomy and distribution of African rodents. And there are many others. I'm finishing. So there are many others by our collaborators in last decade. So it's really time to summarize somehow this information. So we have similar for shrews, but I will not speak about shrews. It's mainly Violan and Adam Konechny who are leading the similar activities for shrews. So in total, we have genetic information of about 25,000 of small mammals. And it's time to revise this book. It was the first attempt to look at rodents of sub-Saharan Africa by Aram Onajem and 3 other co-authors. And we decided in the last African Small Mammal Symposium that we will somehow update this book. So we are working on that, and the submission deadline is December this year. So we still have a lot of things to do, but there should be a really informative picture of skulls and teeth for each genus. There should be phylogenetic trees based on genomic data where they are available. And there should be also distributional maps that will be really reliable based on genotype data or some well-confirmed data. So this is what we are doing now, and it's a big fun, and I like to work on it, but I have not enough time These are the data which are now included in the database, all data, and these are cytochrome B barcoded specimens. So it can be good progress compared to the previous edition of this book. So the last slide. So this will be not only sub-Saharan Africa, it will be complete Africa. So we will include also the Sahara and Northern Africa. And till now we have 499 species. So it will be good to describe one more. So to have 500 species of rodents in Africa. There are some candidates, of course, because there are some genera which are not taxonomically solved, like Graphurus, Dasymes, Gerbillus, large part of Dendromus. So there is a taxonomic mess. And the interesting thing that there is a lack of basic biological information in majority of the species. So for some of species, we have good biological data in some regions like Senegal. So we know a lot of about species of Senegal, but these are just let's say 10 species of these 500. So for majority of species, there is no biological information. And on the contrary, there is a strong focus on 2 or 3 invasive species of African rodents. So if we have the meeting on African rodents, and it's not only here, but it's also this African Small Mammal Symposium. So more than half of talks about African rodents are on house mouse and black rat, which is understandable because they are really making problems to human, and it's probably a bit easier to get money for research of black rats than Dendromus. But it's just how the information on African rodents is biased. So that's it, and thank you for your attention, and thank to many people who helped to work on these topics.